Hello, everyone, and welcome back to ImmunoLogic, the podcast where we splice the cutting edge of immunotherapy into real-world clinical insight with a dash of humor and good measure, hopefully. And I'm Joe Fraietta, your immuno-obsessed host, always on the lookout for innovative ways to help the immune system knock out cancer. You know, we often think of surgery as the trusty, heavyweight champ in cancer care. It literally cuts out the problem. While immunotherapy, I see it as more like a stealthy sort of ninja hunting down any cancer cells hiding in the shadows. Now, imagine if these two could team up, tag teaming breast cancer like an epic duo. So picture a kind of scalpel meets supercharged T cells, sort of a buddy cop movie. That's pretty corny, but it sounds thrilling to me. Well, that partnership is exactly what we're exploring today. But before we dive in, let me introduce my partner in crime, no pun intended, Dr. Janna Minehart. Janna, are you ready to join this? I guess it's a dynamic duo, right, today?
I'm so excited to have our very first surgeon on the podcast
Oh,
with us.
that's true. That's true. Speaking of which, it's such an honor to welcome Dr. Julia Tchou, a dear friend, a mentor, and actually somebody who's getting me into other immunotherapeutic modalities other than engineered T cells. So Dr. Tchou's a breast cancer surgeon, a renowned breast cancer surgeon at the University of Pennsylvania. She's a physician scientist deeply involved in immunotherapy, translational research, and she leads several initiatives and several clinical trials combining surgical modalities with immunotherapy. And her lab at Penn really focuses on understanding how to make breast tumors more visible and vulnerable to the immune system. A couple of hints there. So Dr. Tchou, welcome. We're honored to have you.
Thank you so much, Joe and Janna, for having me. This is my first time on a podcast, so you have to forgive me if I do something that is not right.
No, 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 not at all. We'll keep it fun. So, if it's okay, I'll dive right in. Immunotherapy, it's fair to say, and we've really did belabor this point in other episodes. It's transformed the care for cancers, right? Like melanoma, like lung cancer. But breast cancer has been, I would say, historically, although correct me if I'm wrong, Julia, one of those diseases that's a tougher nut to crack. And for a long time, I think folks considered it immunologically cold, meaning there weren't many immune cells sort of hanging out in the tumor. Early trials of checkpoint inhibitors alone weren't exactly home runs. I think that's fair to say. So why are we seeing so much momentum now? What's the change, and how has the role of surgery evolved alongside this shift?
Oh, such a great question. So the first question is, why breast cancer so cold, immunologically so cold? So the answer is not that simple, because there are hot tumors and cold tumors, and we have to remember breast cancer is a heterogeneous disease. So one of the most notorious breast cancer, the more aggressive type of breast cancer, also known as triple negative breast cancer, is actually not cold. We've seen that there are tumor infiltrating lymphocytes in most of these tumors, ranging from like 10% all the way to 80 or 90%. So about maybe 10 or 15 years ago, I think researchers have been thinking about actually exploiting the immune system to see if we can fight these tumor cells. And that was the start of using immunotherapy. As you've said earlier, monotherapy does not work. There needs to be another immune modulator. In this case, about 15 years ago, chemotherapy, almost like a induction therapy, started to be combined with immunotherapy, which is the immune checkpoint blockade therapy. And they have really good success. And that led to a change in standard practice in the treatment of early stage triple negative breast cancer. But as you can imagine, the combination chemotherapy and immunotherapy for 6 months before surgery is a lot for patients. So you and I have begun to think about a little bit less toxic combination therapy. And this is where we are right now to further explore a chemotherapy-free approach in activating the immune system to kill these tumor cells without the use of chemotherapy.
Yeah, no, fascinating. And for surgeons, this kind of trial is incredibly valuable because, we can directly observe also with surgical resection, perhaps how the tumor microenvironment changes in response to immune activation, which certainly helps us refine, you know, future strategies
Yeah,
as well.
maybe we can talk some more about the details of the I'm guessing this is the RADVAX trial that we're talking about. Is
The
that
breast vax study, yes. Yeah. Can I just go straight into it?
please?
So I think the RADVAX study started the momentum at Penn. In that study, which was completed maybe 10 years ago, Bob van der Heide and his team started it. What they did was they used radiation combined with immune checkpoint blockade, in this case pembrolizumab, to treat more advanced breast cancer. It's known as a basket trial, which means any types of cancer can enroll, but they have to be metastatic cancer. As we now know, immunotherapy works best in earlier stage breast cancer. So in that study, they did not see too many signals coming from breast cancer, but they did see a complete response in kidney cancer, which is a very encouraging. And then borrowing from what we learned from melanoma neoadjuvant study, which means that they're giving the treatment upfront before surgery. And I'll talk a little bit about the role of surgery at this time, because I think Joe talked about how wonderful surgeons are, but I actually think it's the other way around. And, so in melanoma, one single dose of pembrolizumab was sufficient to activate the immune system and also cause a tumor response, tumor shrinkage. In about 30% of the patients, the tumor completely disappear after single dose. So combining these two concepts, we began to come up with the idea of doing this in early stage breast cancer and treating these breast cancer before surgery using low dose radiation. The idea of low dose is to activate the immune system without actually depriving the tumor microenvironment with these immune cells, because higher dose radiation may be toxic to these T cells. So combine low dose radiation plus a single dose of immune checkpoint blockade, which is pembrolizumab, prior to surgery. So we started this study 5.5 years ago during the pandemic, which is another thing that we can talk about. But anyway, so patients, a lot of patients will embrace the idea, but some patients are hesitant, which is understandable. So results are, we have results from 35 patients, and they are encouraging because of the 15 patients or 17 patients with triple negative breast cancer, even after a single dose of pembrolizumab without chemotherapy, we saw about 50% of them having tumor response with 2 complete pathologic response. So which is very encouraging. So we now are going to continue to study this idea, looking at whether we can use this approach to trick the immune system to use the tumor in situ to vaccinate the patient. So it's very cool.
Yeah. And from a surgical standpoint, the timing really matters, but with careful planning, does immunotherapy, what you're talking about, interfere with the standard of care? Or maybe you could comment on that a little bit. Surgical resection.
We are very, very mindful of that, not to delay surgery for this early stage breast cancer. So we designed this window of opportunity study. This window is a very tight window, which is 21 days. We want to give the treatment in this tight window and bring them to surgery. Once they sign consent, the clock starts. So we want to have the surgery be scheduled and done within 3 weeks, which has been feasible in all our patients. So that's a great question. We also know that when we give pembrolizumab, we need to allow about 2 weeks before we do surgery, because it really takes that long for the pembrolizumab systemically to get into the tumor. So we learn from trial and error in a way. In the beginning, if we do the surgery too quickly, the response is lower.
Do you see grossly or pathologically any differences compared to the resection for standard of care?
I'm not quite understanding.
Is the tissue more friable?
Oh,
more
got it.
appearing, or like,
Oh, yeah. Interesting. Very. So actually, I think the tissue feels more scarred, and I think it makes sense, because the scar tissue means that the tissue is reorganizing after the tumor cells died and apoptosis, and the tumor is now being reorganized into a scar, so it feels firmer. Now, it brings me to the next point, which is the lymph node. The lymph node actually looks more enlarged and more activated. They are like inflamed lymph node, just like when you have a cold. So that observation is actually shared in melanoma in my colleagues who did melanoma surgery. So there's something happening in the lymph nodes. That's why I think it's so exciting to be analyzing the tumor as well as the lymph nodes.
That must have given some of you some palpitations at first, if you thought that that meant that the lymph nodes was involved.
Yeah, it does. It makes me wonder, are they cancer-involved lymph nodes, or are they just reactive? So I think it's reactive more than cancer-involved.
That's fascinating. And it makes a lot of sense to give immunotherapy while there's disease present. I think that approach is something that's also understood in lung cancer, where if you giving immunotherapy when you've already resected the disease and those neoantigens may make less sense from an immunologic perspective.
Absolutely. This concept has been tested in melanoma, actually, in a randomized controlled trial. The trial study whether immune checkpoint blockade should be given before or after surgery. And the comparison arm is pembrolizumab before and pembrolizumab after, and they saw a significant survival difference favoring pembrolizumab given before surgery. And that makes sense, right?
Right. Yeah. It's nice when things follow our biological paradigm.
Very rare for me.
Yeah.
No, but that's so exciting. The other thing is, after standard of care, which is removing a tumor, surgeons and oncologists alike really worry about, what we've touched on in a prior episode, which is microscopic residual disease, right? The cancer cells that we can't see, but that could certainly lead to recurrence. So do you think your strategy is maybe superior to standard of care, which is surgical resection followed by chemotherapy for higher risk patients? I'm just wondering if you could unpack that a little bit.
Yeah, I think minimal residual disease is really something that we have to spend more time doing research on. So whether treating upfront with a little bit of radiation plus immune checkpoint inhibitor is helpful or not to minimize the risk of residual disease to emerge in the future is unknown. We think that it will be beneficial because as we imagine the tumor serving as a vaccine, there will be epitope spreading. The immune system is now poised to be recognizing the tumors of the heterogeneous pool of tumor antigens, right?
Right.
But this has not been tested, and we would like to test this in the future in the next breast FACS-2 study. And so stay tuned. Happy to come back in a few years.
Oh, yes. Sooner than that, we hope. Are you thinking, though, in that vein of, I guess it's too complicated for these, initial trials, but patient stratification up front to see, who might benefit better from sort of the breast FACS approach versus the standard? How are you and your colleagues thinking about that?
So all these patients that were eligible in breast FACS study would not be eligible for upfront chemotherapy. That's why we wanted to have these patients enroll in the study. So we don't want to move them off standard of care. So so the question is, how do we think about patient eligibility in the next study? Again, we have to be mindful, not deviating from standard of care and not depriving patients from the treatment that has been proven to be life savings, right? So the next study would be focusing on triple negative breast cancer stage 1. And we are hoping that the combination single dose of radiation plus single dose of pembrolizumab will activate the immune system and cause a major pathologic response. And then we're going to start selecting patients or having patients make a shared decision to omit chemotherapy altogether if they have a major response, and then also giving them an option to continue several more doses of immune checkpoint blockade to ensure that the immune memory is durable. So it's a de-escalation surgery, but de-risking the de-escalation part. Does that make sense?
Yes, very much. Fascinating concept, I think. We're really thinking about it differently these days, and bringing immunotherapy and these combinatorial approaches more to the front line, right?
Mm-hmm.
Which I think is fascinating. Now, I have to do it. Janna will yell at me. Engineered immune cells, because I've been following also a lot of your clinical and preclinical work, because you've been a PI on some CAR T trials. But a more recent publication that I was looking at just last night was applying engineered immune cells directly into the CAR T cells, directly into the surgical cavity, with the idea of dramatically reducing recurrence, like we just talked about. So I guess the idea is to leave behind an immune system or immune security system. I'm trying to tie back to my corny intro, right? That immediately attacks any of those residual cancer cells. So is that moving into trials, or how are you thinking about that?
So I think Carl and Yugi did that study along with, I'm one of the co-authors. That idea came from, actually, I think Carl and I thought about the idea together. So, I think the idea is great because using the residual tumor, if there's any, and then applying the engineered T cells into the cavity itself will accomplish a couple of things. One is getting the T cells into the tumor itself, and second is decrease the off-target effects, right? And then and what we hope for is, if there's tumor cells there, the engineered T cells will recognize it and start having epitope spreading, which is the ultimate goal. So whether we can do this in breast cancer is still early, because you have to think about the population. I think the T cell manufacturing takes time, and then we have to think about timing and think about the population. If they are too advanced, if they have advanced breast cancer, we usually don't do surgery. So it's like a niche population that we have to think about. And I think it can be done, but we have to identify the tumor target. Can I talk about tumor targets?
Please. Yeah, it's great.
So we did the mesothelin targeted, mesothelin directed CAR T cells, one of the early study we targeted c-Met first, but c-Met was targetable. However, we did not see any tumor response. But that's using an RNA CAR construct. We moved on to mesothelin, a lentivirus construct. As you've done some of the analysis, we see persistence of this CAR T cell in the blood, but we also see very minimal response. The problem is mesothelin as a target for triple negative breast cancer. It's thought to be a good target, but when we screen patients, we have less than 1 in 10 patients eligible. So
yeah,
we think that the mesothelin in breast cancer may have been, it's a surface membrane protein. It might have been cleaved off, so we cannot find it. So we have other targets collaborating with the Netherlands. We have another target coming up. It's an engineered T cells, not CAR T cell. So an engineered TCR, not the CAR construct.
oh, got it.
Yeah, so it's targeting a embryonic antigen. So it seems promising. So we are in discussion with that, opening it in the United States.
And
Oh,
would that
wow.
intratumoral delivery, or that would be
Well, that remains to be determined. Of course, these company wants infusion, right? Because intratumoral injection is probably the safest way to do it initially. But I think from the company standpoint, they want to develop a product that is systemic or infusion based. I don't know why, but maybe
easier to operationalize.
yes, yeah. But I'm going to meet with them and talk about the study design in 2 weeks. So more to come. It's exciting.
That is really exciting. It's funny as you're talking, I'm thinking about lymphoma because I always do. We think
Like I always think about CAR T cells.
yeah.
You can't help it. You have to bring lymphoma in.
Yeah. Well, in lymphoma, we think of breast tissue as immune privilege site, and that patients who have breast involvement or testicular or adrenal, these are folks that are at higher risk for higher risk disease and higher risk of developing secondary CNS lymphoma. And I think that in my biology education, I had thought of the testicles as an immune privilege site. I've thought of the CNS, but I never really thought about the breasts being so until I learned that about lymphoma. But it makes some sense, I think, then too, why so many breast malignancies are immunologically cold, perhaps, and also maybe perhaps a justification for that. Intratumoral or in the resection cavity kind of delivery if we think that there might be some issues with trafficking specifically to breast tissue.
Yeah. I think trafficking is one of the issues into the breast tissue itself. So the intratumoral injection delivery space, I think it potentially can apply to pancreatic cancer, which is a much more deadly disease. So I think Bruce Levine and Carl and I thought about a paintable CAR T cell, paintable. So when they do pancreatic surgery, they cut the pancreas in half, and then they left the half to and sew it up to the bowel so that the pancreas can continue to produce enzyme and whatnot. So
that's the Whipple.
Yeah, that's a Whipple.
Yeah, I knew that.
Am I describing it too late?
No. I mean, I just wanted to throw that in. That's
a Whipple procedure.
He wants to know that he's begun his medical school education.
Exactly.
Yeah, this is all we do. We cut it in half and then sew it back to bowel. So I was thinking that if this is a paintable CAR T cell, you can just paint it onto the cut edge of the pancreas. And then the CAR will stay for a while and do its thing. It'll be great, right? But I'm not quite sure if the pancreas surgeon. I don't do pancreas surgery anymore. I don't know if this is gonna have any traction or not. But I think it makes sense, right?
Definitely.
Especially since we know that there's some sequestration of CAR T cells, like, particularly in the pulmonary vasculature when they're administered systemically. And I think there's some thought that maybe that delay before they're then able to ultimately find their target might hinder their activation and activity.
Right. We can paint it on.
Right.
Yeah.
Then it's right there. It can find its
Yes.
target and
Yes. Why not? Yeah.
be activated without delay.
So we actually used surgical glue of tissue glue, which is like a biologic that we use all the time. It's called Tchou. It's just clotting factors. It will and thrombin. It forms a little gel that you can just apply on the surface. It sticks on and stay for maybe 6 hours or so. So long enough for them to kind of seep into the pancreas.
That's a fascinating.
Nobody took my idea, I don't think.
Until they watch this, right?
Yes, pancreas surgeon.
Yeah.
Take note.
Right. So I mean, beyond CAR T cells, we're moving toward personalization also in the vaccine space as well. So beyond the in situ vaccine approach, which I like because it's simple, and I think you told me once you had a number or a few patients where you were able to do the subablative dose of radiation and actually give the checkpoint blockade on the same day. Um,
Yep.
so I really love that for logistical reasons. But do you think that's enough, or do we need more personalized neoantigen sort of like vaccine strategies? Maybe you have to do that, and then I don't know. I'm just thinking of your trial schema
Yeah.
sequence, and then
Yeah.
maybe make a custom sort of mRNA vaccine. Just have you
Yeah.
thought about that, or is
I,
it too much, too complicated, not feasible?
no, I think there's I just came off of a 2-day think tank conference
Really?
of Cancer Vaccine Coalition, com, uh, kind of a AACR, joint with the Cancer Vaccine Coalition think tank discussion. So I think the consensus is intratumoral delivery of vaccines in any form, peptides, RNA, DNA, virus, plus an immune modulator is necessary to
Mm,
activate the immune system. So, and then I think from learning from everyone, I think the immune modulator needs to be customized or personalized because it may not work in all types of cancer or individuals. So um, so I think we learn a lot from the combination of RT plus immune checkpoint inhibitor. I think that's just the beginning. So going forward, so when we think about cancer vaccine, there's a difference between prevention versus
mm,
for cancer therapeutics. Prevention needs to be off the shelf um, to be implemented. Um, Cancer vaccine, therapeutic vaccine, I think it needs to be customized or personalized. Um, so, so, uh, and I think the approach that we take as the breast FACS is probably one thing, a study schema that we can learn from, right?
sure.
So, yeah, I think surgeons can lead this effort, especially in the space of pre-malignant and early stage cancer, and then maybe prevention as well. So, but for the later stage, I think we still have a role, but I think it will be more of a role of the oncologist. So, plus
Got it.
surgeon.
Got it.
We
Well,
we work together
yeah,
well.
yeah, definitely, right? You need hence the dynamic duo, right? That I was talking about.
And
um,
you,
yeah,
dynamic trio.
wherever I fall in the lab somewhere, right? But Dr. Tchou, thank you for such an insightful and inspiring conversation. I love it. Usually when we meet, you're post-op in scrubs, and we're zooming that way. So and that would have been okay if you did that today too. But you've really shown us how surgery and immunotherapy aren't competing tools. They're maybe the best collaborators out there. And the future, I think, really looks bright in that space. So, thank you. It's really been a pleasure, and I'm excited about where this field is headed.
Thank you
And
so much for having me.
yeah, thank you. And to our listeners, thank you for joining us for this deep dive into the future of breast cancer care. And until next time, stay curious, stay collaborative, and stay immunologic.